So I work at Yale School of Medicine in a neurobio research lab, and here's how Claude Code helped me draft the entire results section for one of my experiments in under an hour. This is a section of a mouse brain that has two different proteins stained in red and green, and I needed to count how many cells express the red protein and how many cells express the green protein. To do this, I had Claude Code write me a script for an image editing software called Fiji, which if you're a scientist, I'm sure you've used before. And essentially the script created a grid on top of the tissue section, and it used the brightness of the pixel values in the picture to count how many cells stained positive for red or green proteins within one grid square. Then I prompted Claude Code to extract the data from Fiji with a Python script and format it nicely into a table for easy data analysis. Then I prompted Claude to perform various different types of statistical analysis on my data to see if there were any significant differences in how the proteins were expressed across the tissue. It then compared the results of those different statistical models and came up with the one that yielded the most significant results, and it generated all four of these beautiful plots to me to convey the results. After collecting, compiling, and analyzing all of my data, I can now talk with Claude about how it would be best to display this data to people trying to understand it. These plots were completely generated by Claude, of course with my instruction, and as you can see, the top plot clearly shows that the protein labeled in red is expressed more at the left side of the tissue than the right side of the tissue, and the protein labeled in green is expressed more on the right side of the tissue than the left side of the tissue. You can also see a clearly labeled breakpoint where the trend in the protein expression changes, and that results from the statistical analysis that Claude performed. And here's the two charts overlaid next to each other, representing the protein expression of both proteins across the entire tissue. Because most of the code that did the data collection, extraction, and analysis was created in my conversation with Claude, it knew the entire context of basically all of the methods that I used to collect and display the data. I then copy-pasted the entire chat history into a document in a Claude desktop project and also added a bunch of background research papers from existing literature on the topic to give Claude some context in helping me draft the research report. Based on all this information, Claude drafted me a complete and entire method section for this figure in my paper. Now, obviously, none of this will actually be used in the publication because that would be fucking crazy, but it basically allowed me to structure my entire experiment, or at least this figure, in a paper based off of the scripts that I wrote with Claude and based off of existing background literature. And just for final clarification, none of these scripts, the charts, or the text that was generated in this conversation will even make it to a draft of the paper, let alone the final publication. But it essentially allowed me to put together the entire idea for a figure in a paper and show it to the other members of my lab, discuss it, talk about issues, talk about good things, without having to do much work in actually performing the data analysis myself. If you're a scientist and you're not using tools like this, or you just don't know how to use tools like this to do your work faster and more efficiently, I have a school community that teaches people how to use AI tools like Claude Code to accomplish any work, whether it's coding, whether it's business, or whether it's science, more efficiently and more quickly with tools like Claude Code. If you're interested, the link is in my bio. We would love to have some more scientists